Okay, so in this video, we'll be talking about fatty acid oxidation. And I want you to be able to know the reactions catalyzed by acyl-CoA synthetase and carnitine acyltransferase specifically. Where and how do fatty acids become activated for this beta oxidation? Where does beta oxidation occur? I want you to be able to recognize the pattern of oxidation that occurs to these fatty acids. You don't necessarily need to know the specific enzymes, but there's a pattern of oxidation, and we'll go through that in a moment. What are the products of one round of beta oxidation? And what is the final product of the beta oxidation of odd numbered fatty acids? This is something special. And what familiar intermediate can this odd numbered product be converted to? All right, so first, the 3 steps of fatty acids is first we have to activate the fatty acids that have been released from the glycerol molecule, and that occurs within the cytoplasm. They then need to be transported into the matrix, which is where beta oxidation is going to occur. So activation is catalyzed by an enzyme called acyl-CoA synthetase, and this depends upon an adenylated intermediate. So not a phosphorylated intermediate, but an adenylated intermediate, which means that what we're spending is an entire ATP. So we're getting rid of one phosphate, we're getting rid of 2 phosphates, so we're left with a byproduct of AMP, getting rid of an entire pyrophosphate molecule there. And in so doing, what we end up with is this activated, what we call a fatty acyl-CoA. So R is our carbon chain, right, between 4 and 30 or so carbons long. And then this used to be the carboxyl group at the end of the fatty acid, but it has been activated by being attached to a CoA. So we've got that thioester bond again. So another example of a thioester bond. Now, in order to get that fatty acyl-CoA from the cytoplasm into the mitochondrial matrix, we need to take use of an enzyme called the carnitine acyltransferase that substitutes that CoA for a carnitine molecule. And now this carnitine fatty acid can move through this carnitine transporter, where then within the matrix, this fatty acid that has attached to carnitine is then replaced, that carnitine is replaced by a CoA again. So here is now our fatty acyl-CoA. Okay, so again, we're moving our fatty acyl-CoA from the cytosol to the matrix by first adding a carnitine to it, moving it through a carnitine transporter, and then adding the CoA back to it. Once we have our fatty acyl-CoA within the mitochondrial matrix, beta oxidation occurs. And the way it occurs is that starting from the carboxyl end, every 2 carbons, so 2 carbons at a time, are clipped off as acetyl-CoA. And the process by which that occurs itself releases 1 NADH and 1 FADH2, in addition to all of the NADHs, all of the FADHs, and all of the ATP that the cell gets from oxidizing acetyl-CoA within the citric acid cycle. And so every 2 carbons in acetyl-CoA, every 2 carbons in acetyl-CoA. So let's take a look at beta oxidation, and let's take a look at the pattern of this oxidation. Now, as a reminder, you will not have to memorize any of these enzymes, but what I do want you to memorize is the pattern of oxidation. So let me explain what I mean here. So here is our fatty acyl-CoA, right? There's the CoA there. This is that last carbon, right? It used to be that carboxyl group of the fatty acid. So the carbon after that is the alpha, and then the third carbon is the beta. Beta oxidation, we are oxidizing this carbon right here. Okay? Which means this carbon right here is going to end up being, again, within a thioester bond connected to a CoA. So let's take a look at what happens here. First, we have an alkane, which becomes oxidized to an alkene. Okay, we have oxidation happening. We're removing two hydrogens and their accompanying electrons. We need something with enough oxidizing power to do that. There's our FADH2 right there. And then we oxidize this again. We oxidize it from an alkene to an alcohol. Okay? And then we take it from this alcohol and we convert it into a carbonyl. Now, going from an alcohol to a carbonyl, that's going to take an NAD plus, becoming an NADH. So there's our one FADH2 from this process. Here's our one NADH from the process. And then finally, we have a thiolysis reaction. We're using this sulfhydryl group. Remember, thio is sulfur. We use this sulfhydryl group to attack this carbon right here and form that thioester bond, and that lyses this bond here between the beta and the alpha. So we lose this as our, or I guess we gain that really as our acyl-CoA. And here is our new end of the fatty acyl-CoA that's been reduced by two carbons. We went from C16 to C14, and here's our acyl-CoA. So I want you to be able to look at this: alkane to alkene to alcohol to carbonyl to our thiolysis, resulting in our fatty acyl-CoA reduced by two carbons and and an acyl-CoA on the side. Now, what I have here in red is I'd like you to look at these reactions here: alkane, alkene, hydroxyl, carbonyl. You've seen exactly these before. I want you to go back, look at your notes, think about this. Where have you seen this exact same sequence of reactions? I'm not going to give you the answer here. Now, what happens when beta oxidation is working on a fatty acid that has an odd number of carbons? Well, the answer is beta oxidation ends with a 3-carbon molecule, Propionyl-CoA. At that point, it's not going to go any farther. What happens is there's a set of reactions in which this Propionyl-CoA, this 3-carbon molecule, is converted into succinyl-CoA. Now, you don't have to memorize any of these enzymes that do this. You just need to memorize that Propionyl-CoA, the product of odd carbon fatty acids, is converted into succinyl-CoA. And from there, we all know where we're at. We're at a familiar intermediate of the citric acid cycle, and you know everything about succinyl-CoA there and what it can be involved in. Now, I have again this red question. Based on this information, based on the fact that only odd numbered carbons can give rise to succinyl-CoA, can give rise to an intermediate of the citric acid cycle, and all of the other even-numbered fatty acids, they can only give rise to acyl-CoA. There's something special that odd-numbered fatty acids can do that the evens cannot. And again, I want you to think about that, and we'll talk about that in class. All right, So again, make sure that you can go through each one of these and answer these questions. Make sure you think about those red questions that I have you thinking about, and be prepared to discuss those in class.